Welcome to Metabolism and Bioenergetics Part 3. In this tutorial, we'll look at both the electron transport chain and oxidative phosphorylation. Because they're so closely linked, it's really hard to separate them. Let's get started. Alrighty, so this abbreviation we've already talked about, but just to make clear, right, ETC will be the electron transport chain. And this is all about the reduced coenzymes. Right, transferring the electrons so that we can get ATP molecules. And then the oxidative phosphorylation, right, that's where we have the ADP and we have oxygen, and then we can bring in some hydrogen ions, and this is where we finally get our ATP and some water, right? So the reduced coenzymes you can think of as stored energy available, but ATP is the readily accessed. That's when we have energy in an active form that we can use. So let's look a little bit about where all this is happening inside our cells. So the mitochondrion is the organelle where the chemistry is going to be happening that we're talking about in this chapter. So very briefly, right, we'll look, so there's the cytosol, which we know is the fluid portion of the cytoplasm. Okay, so that's where everything floats around, and then where we really want to focus is right here, the mitochondrion. And amazingly, 90% of energy production takes place in this organelle. So very important. Now this organelle has a very complicated structure, but we need to understand it to be able to understand the citric acid cycle and the electron transport chain. So I feel like this schematic does a good job of helping us keep our bearings. So the mitochondrion has a double membrane layer. Right? So we have this outer membrane, and then there's the inner membrane. Okay, and then then here we can see the cross section, right? So here's the outer and the inner membrane, and we see how the inner membrane folds in and creates all this surface area. And then there's also this gap in between. So then there's the space between the two membranes, the intermembrane space. Okay, so now if we look at the inner membrane, we can see there's the ATP synthase enzymes, and so that will be the sites for ATP production. And then on the very inside, in the innermost area of the mitochondrion, we call that the matrix, right? So that's the innermost region of the mitochondria. All right. Okay, so now that we have our bearings of where, because we will be talking about the inner membrane and this intermembrane space and the matrix, right? Because this is where the enzymes occur. So the matrix, the inner membrane, and this intermembrane space. We will be talking about all of that as we proceed through this tutorial. So it's important to know where we're talking about. Okay, so now we have to look at a bit of the energetics before we can get to the actual energy production. So what's going to happen in the electron transport chain is that the electron transport chain is going to create a proton gradient. And this proton gradient creates something we call the protomotive force. Basically, it creates stored potential energy. All right? So we'll look at it conceptually, and then we'll get to the nitty-gritty details, right? So the ions dialyze through the membrane in a direction that equalizes the concentration of ions, right? So that's the normal flow, high concentration to low. So ions are unable to pass through the inner membrane by simple diffusion, so that creates the proton concentration differences on either side of the inner membrane. And so the matrix has a lower concentration of protons than the intermembrane space. So what we're going to see is that the electron transport chain is going to pump protons into this intermembrane space. All right? And so that creates this concentration gradient. So let's touch base with what we've learned about acid-base chemistry and pH. So if the matrix has a lower concentration of protons compared to the intermembrane space, is the pH lower or higher? Right? So remember that inverse relationship. A lower concentration of protons means a higher pH. And so then the intermembrane space has a higher concentration, right, because they've been pumped in. So there's a higher proton concentration. So remember that that leads to a lower pH, right? Okay, so let's take this explanation and see, right? So we can remember here is our mitochondrion, right? So we know that the matrix has the higher pH, and the intermembrane space has the lower pH. So now if we look at our proton gradient, we want to find out which part is representing the matrix and which part is representing the intermembrane space. So we would probably draw our line right here. Right? So the matrix would be represented here, right? Higher pH, lower H plus concentration. And then this part of the diagram would represent the intermembrane space, where it has the lower pH, right, and the higher hydrogen ion concentration. And so that would be, right, higher pH. And that's talked about over here. Okay. So now that we've got those pieces put together, let's do a little wrap up here on this page, right? So most of the enzymes involved in the citric acid cycle reside in the matrix, right, or along the inner membrane. Well, for the citric acid cycle, they're inside the matrix. For oxidative phosphorylation, we saw they're on the inner membrane. So the protein complexes involved in, okay, I just talked about that. So the important enzyme ATP synthase extends from the intermembrane through all the way into the matrix. And that was shown on the previous page. Alrighty. So now let's get to the heart of the electron transport chain. So here it is in words, and then we'll look at diagrams to help explain it. Alrighty. So, right, the electron transport chain takes the electrons from the reduced coenzymes and passes them through a chain of cofactors and enzyme complexes that result in 4 electrons and 4 protons connecting with oxygen to form water. As the electrons are transferred, the electron transport chain pumps the protons into the intermembrane space. Since the movement of the protons is against the proton gradient, the process is referred to as a proton pump. The accumulation of the protons in the intermembrane space represents stored chemical and electrical potential energy, which creates that proton or protomotive force. All right, so that's the key. It's like winding up the spring or the engine, right? Or charging the battery. Okay, So now let's look at it schematically. All right, so the first thing to note is that the NADH enters at complex 1, and so it transfers the electrons, and so then it becomes oxidized. Notice that complex 1, 3, and 4 go all the way from the matrix through the intermembrane. Complex 2 receives the electrons from FADH2, and so then it becomes oxidized to FAD, and the electrons are transferred to complex 2. So with complex 1, we get protons pumped, and complex 3, we get some more protons pumped. Notice complex 2, since it does not penetrate all the way through the inner membrane, there are no protons pumped from complex 2. Now complexes 1, 2, 3, and 4 are rigidly stuck in the inner mitochondrial membrane. So we have coenzyme Q and cytochrome C. So these are the mobile proteins that will help to facilitate the transfer of electrons through the chain. Alrighty. And notice that what's happening then is the protons are being pumped into the intermembrane space. So we have the high proton concentration here, and we have the low proton concentration here, right? So this is what sets up the gradient, right? So the protons, they want to diffuse from high concentration to low concentration, right? But we have this non-polar membrane creates the barrier. Right, so this creates the barrier to dialysis of the ions, right? So that's where we'll think about complex 5, right? So that's why we're going to need the ATP synthase. And we'll look at that very soon. And so we think of that as complex 5. So there's a natural tendency, that's what creates the protomotive force. These protons want to diffuse to create a uniform proton concentration, but the non-polar membrane is creating a barrier. And so that's what creates the protomotive force. Right, oops, right there, protomotive force. Okay, so then energetically, let's look at this a little bit more before we get into the details. Right? So if we look energetically, right, so we have the NADH has the highest energy, and we look at water as having the lowest energy. All right, so these different complexes, right, so complexes 1, 2, 3, and 4, right? They are complex, right? So we're going to simplify them. It's important to realize that they have multiple enzymes, right? They are multi-enzyme protein complexes. So we are definitely having some oversimplifications. And so then we can see, right, that NADH is willing to lose its electrons. So we would say that it has the lowest electron affinity because it's willing to give up its electrons. And so then we would look at, right, or NADH, and then even amongst the, so this is about the complexes though, right? So complex 1 has the lowest electron affinity, and complex 4 has the highest electron affinity, right? And so affinity just means, right, you have an affinity or a like for something. So complex 1 is willing to let go of its electrons, saying it has the lowest affinity, and complex 4, it wants them. Okay, now before we get to the full details of the electron transport chain, let's look a little more closely at these mobile proteins, the coenzyme Q and the cytochrome C. And then we'll have all the pieces in place to talk about the full electron transport chain. Okay, so alrighty. So the coenzyme Q and the cytochrome C are important because those are the mobile proteins that are needed for the fully functioning electron transport chain. So coenzyme Q is also called ubiquinone because it is so ubiquitous within our body. And so we're not going to, well actually we're going to look at the whole structure here. You can see it's not too bad. We have our quinone structure here. So we have some ketones and some ethers and a hydrocarbon tail with a double bond. And then we can see here's coenzyme QH2. So if we were going, right, so we would call this CoQ, and then we would call this CoQH2. So let's look for the changes that are occurring and recognize which form is the oxidized form and which form is the reduced form. So as we compare and contrast the different structural features, we notice that we have ketones here and we have alcohols here. And then notice the formation of the aromatic ring. All right? So if we look at these two molecules based off these structural differences, we recognize that coenzyme Q is the oxidized form, and that the CoQH2 would be the reduced form. And that's not too surprising because we've come to associate hydrogens with the reduced form. And so we see there are more hydrogens present. So then we could look at our reaction arrows here, and so we could say that the forward reaction is the reduction reaction, and the reverse reaction then, of course, is the oxidation reaction. All right, so this is happening, right, as the coenzyme Q, right, right, so it accepts the electrons. Maybe that should be green, right? So it accepts the electrons, right, as part of the electron transport chain and becomes CoQH2, and then the reduced coenzyme Q can then hand the electrons off to another protein complex in the chain. So now we'll look a little bit at cytochrome C because we can see that coenzyme Q, this is an organic-based oxidizing and reducing agent. However, with coenzyme Q, we have to touch back onto our inorganic chemistry, right? Because we see that it's the iron at the center of cytochrome C, which is a protein, right? And this is a heme group. And so it is the iron that accepts the electrons, right, as part of the electron transport chain, and then is able to pass them on. So if we look closely at cytochrome C, we see here we have iron 3+, which we would say is the oxidized form, right, because it has the least number of electrons. And then as it gains electrons, right, gaining electrons is reduction, right, so electron gained. So then this would be our reduced form. And of course, fortunately for us, we wouldn't be here to talk about it. It can oxidize back and forth. And so we see this in the electron transport chain and also in our blood with hemoglobin. Alrighty. So now I think we have all of the pieces that we need to go through the actual chemical process of the electron transport chain. So let's put it all together now that we have all the pieces. Okay, so keeping in mind where this is all occurring, right? So it's occurring on basically everything's happening on the inner mitochondrial membrane, in the matrix or the intermembrane space, right? So, and we have our potential gradient. We have, because the protons are pumped into the intermembrane space, it has the lower pH, and we have the higher pH in the matrix. Okay, so let's go through. So as we can see, right, the NAD, right, complex 1 accepts, right, 2 electrons from NADH, all right? These electrons are then passed to coenzyme Q, and we get 2 protons pumped into the intermembrane space. Okay, so accepts 2 electrons from NADH, which are passed on to coenzyme Q, right? So then, and then we get we get the pumping of and pumps 2 H pluses into the intermembrane space. Okay. And then we notice, right, complex 2, right? So notice that complex 1 goes straight to coenzyme Q and complex 2, right? So it's not a sequence, right? So complex 2 accepts 2 electrons from FADH2, all right, which are also passed on to coenzyme Q, right? Notice that there are no hydrogen ions are pumped, right? Because coenzyme Q doesn't penetrate into the intermembrane space. Okay, so now the paths converge. Coenzyme Q, regardless of where it receives the electrons, is going to pass them on to complex 3. All right, so complex 3, right, accepts the electrons from coenzyme Q, right, and transfers them. It's just a very elaborate like relay, right, and transfers the electrons, right, to cytochrome C. And there's several cytochromes out there, so we're definitely, oops, excuse me, I didn't slide this up, right? So there are several cytochromes. We're just going to really simplify. The important thing to notice is we get, right, so additional H plus ions are pumped into that intermembrane space from complex C. All right, and that's done right here, right? So we see that complex 1, complex 3, and complex 4. Oh, except that looks weird. I think that this is a mistake. This arrow should point in, right? Because there are the hydrogens. Okay. Now let's look at the last complex, right? So complex 4 then, this is where the cytochrome, right? So complex 4, right, accepts electrons from cytochrome C, right, and produces, right, and produces water. Okay, and the reaction is shown right here. Okay, now, so there is the main, so we've pretty much covered the main elements of the electron transport chain. cyanide, it's kind of notorious, right? The spies carry the cyanide capsule in their mouth, right? So because they don't want to divulge the secrets. So how does cyanide poison us? It's very interesting. It irreversibly binds to an enzyme in the complex 4 system that stops the electron transport chain. So basically it's irreversible inhibition. When something irreversibly binds to one of our enzymes and inhibits it, it takes it out of play. And without the electron transport chain, we're going to die because we don't have any energy. So it's an irreversible binding, right, to an enzyme in the electron transport chain, okay? And so the main thing, right, so causing inhibition, that basically, right, and this is inhibitory. Oops, running out of space here, right? So this is inhibition. And because it's irreversible, basically it stops the electron transport chain. There is a treatment. If you're exposed to cyanide poisoning and you're able to get to a hyperbaric oxygen chamber, we can use Le Chatelier's principle as a treatment to reverse the effects of cyanide poisoning. But the main thing here is just to think about poisons, in many cases, irreversibly bind to our enzymes in an inhibitory way. So wanted to bring in that connection for you. And then we have one last page, I think, yes, one last page where we'll look at where do we actually get the ATP from? Well, that will be with the ATP synthesis, right? So the main thing of the electron transport chain, right, is that it has created, right, the protomotive force, right, by pumping all of those protons into the intermembrane space. So the ATP synthesis, in some biochem books, they actually describe it as complex 5, all right? So all of the reactions in the electron transport chain are energetically favored and use up the energy stored in the reduced cofactors. So but this energy must be saved to make the ATP. And that happens through the creation of the pH gradient or the proton gradient, which is a very high energy state. So those high energy reduced cofactors, their energy is transferred to this high energy state of the proton gradient. And so then what happens is, because the ions have a natural tendency to want to diffuse from high to low concentration, but there's only one way for them to get there. They have to go through the ATP synthase, right? So here we see our proton gradient. Those protons want to get back into the matrix, but to get there, they have to pass through the ATP synthase, or what we could think of as complex 5. So now we'll look at this region here. We have a blown up schematic of this portion here. And so that we see as the protons enter ATP synthase, it's like water wheel turning. And so it provides the energy for ADP and the phosphate group to form ATP. So there's our energy currency. And so then I think that's basically all I want to say there, right? So the net result is we have ADP plus PI, and then we get ATP plus water, and basically this is fueled by the protomotive force, right? The potential energy of the gradient, and then that energy gets transferred from the potential energy of this gradient into the energy held in that phosphoanhydride bond, and so that's our energy currency. Okay, so that wraps up the electron transport chain and oxidative phosphorylation. Please take time now to work some homework problems to reinforce your understanding.